Dear colleagues, composition of semen microbiota in patients with infertility is of great interest for practical medicine. A number of methods are used for semen microbiota composition assessment. The real-time PCR enterofluor test was introduced recently along with traditional culture techniques. This method allows identifying all participants of complex microbial communities, including non-culturable microorganisms. The aim of the study was to compare semen microbiota composition analyzed by means of culture-based technique and real-time PCR-based enterofluor test. For these purposes, semen samples of 92 men were analyzed simultaneously by means of cultures and real-time PCR. Real-time PCR was performed using the enterofluor real-time PCR kit produced by DNA Technology Russia. The kit allows detecting 24 groups of bacteria, including gram-positive and gram-negative bacteria, obligate anaerobes, mycoplasmas, lactobacillus species, as transient microbiota of man's urogenital tract. Once the amplification was over, the special software was used to automatically calculate the total bacterial load and the proportion of particular groups of bacteria in relation to the total bacterial load. Semen cultures were positive in 79% of all samples. So 21% of all samples were culture negative. The growth of a single bacterial culture was established in 34 samples. 2 bacterial cultures were identified in 27 samples. 3 groups, 3 cultures were detected in 9 samples, and 4 cultures in 3 samples. So 28 bacterial species, mostly gram-positive and gram-negative facultative anaerobes, were identified. Bacterial DNA was identified in all 92 semen samples using real-time PCR. And from 8 to 15 groups of bacteria were detected in each sample. The proportion of each bacterial group in total bacterial load was calculated. The predominant group of bacteria, the proportion of which in the total bacterial load exceeded that of other bacteria detected, was determined in the most samples. According to the predominant group of microorganisms, 6 types of semen microbiota were discriminated. Their detection rate is presented at this slide. Real-time PCR in all cases confirmed culture findings. However, in all culture-positive semen samples, other microorganisms were identified by PCR. Mostly those were non-culturable or difficult to culture species. Next, we analyzed the concordance of the determined predominant group of bacteria by culture method and by real-time PCR. The culture results matched those of real-time PCR in 24% semen samples. In these samples, the only isolated species or the quantitatively predominant species detected by culture method belong to the same predominant group as detected by real-time PCR. In other cases, either culture was negative or real-time PCR determined other groups of bacteria as predominant. So culture technique failed to reveal the majority of microorganisms in the samples. Moreover, every 6th sample was considered culture negative. Using real-time PCR, from 8 to 15 bacterial groups were identified in all samples. Real-time PCR established the predominant group of bacteria in most samples. Additional species other than those detected by culture techniques were registered in all samples. As for the predominant bacterial groups, the culture results corresponded those of real-time PCR in only 24% of cases. And the discrepancies were mainly associated with the culture techniques' inability to detect difficult to culture or non-culturable bacteria, whereas enterofluor test allows detecting such. Thank you for your attention.